So hello everyone, welcome to our next lecture. So today we're going to be looking at our antiviral innate immunity. So just a quick overview. Our key innate defenses against viruses include our natural killer cells, our NK cells, and the cells of the type 1 and type 3 interferons. So we use PRRs, or pattern recognition receptors, which we discussed last time, mediated recognition of viruses components, and this results in the activation of macrophages and dendritic cells, and to induce interferon production. So we'll see this IFN, which is interferon, all throughout this lecture. So interferons are important in antiviral defense. So several PRRs are used for detection of viral nucleic acids to induce interferon production. A common goal of the activation of interferon regulatory factors that regulate interferon gene expression. So this kind of happens regardless of which interferon is used. As we'll see, there are different types. Interferons act through interferon receptors, or IFN-Rs, on neighboring cells. This results in signal transduction and transcription factor activation, and this transcription factor is ISGF3, or interferon stimulated gene factor 3. This results in interferon stimulated gene expression, or ISG, and this results in antiviral proteins and actions. And we'll discuss these actions and proteins later. So we have a few different categories for interferons, but first we'll talk about the type 3. So the type 3 interferons also have antiviral activity and include different types of interferon lambda. So type 3 interferons and type 1 interferons bind to different receptors. Type 3 uses a heterodimeric cytokine receptor complex, as we can see here. IL-28 are receptor alpha and IL-10 receptor beta. And type 1 uses interferon alpha receptor. So this big A is actually alpha. These both have similar outcomes. They both activate ISGF3, which is the transcription factor complex, and this results in nuclear translocation, and this activates the transcription of ISG, or interferon stimulated genes. There's also a difference in receptor distribution. So the type 1 interferon alpha receptors are widely expressed on most cells of the immune system, while type 3 receptors are mainly found on epithelial cells and have roles in antiviral responses at epithelial surfaces, like our intestines and mucosal surfaces in our airways. So again, we can see here signaling by the type 1 interferons through interferon alpha receptor, and type 3 interferons through the cytokine receptor complex here, IL-28 and IL-10, induces assembly of transcription factor complex, ISGF3, from STAT1, STAT2, and IRF9. This translocates to the nucleus to activate ISG expression and antiviral protein production. So antiviral protein is also written as AVP, and we'll see some of these later. So again, the interferons. So the type 1 interferons include interferon alpha and interferon beta. These are cytokines that interfere with viral propagation. These are expressed and secreted by a variety of cell types in response to virus infection. The type 1 interferons then bind to interferon receptors on neighboring cells, called interferon alpha receptor, or interferon AR, which induce an antiviral state. This results in virus synthesis being inhibited and the spread of the virus is blocked. So here we can see a cell in A without interferons. So essentially, we have a virus-infected cell, replicates, goes to the next cell, replicates again and again and again. Without interferon, it would just continue. So here we have interferons. So we have our initial infection, which sometimes cannot be avoided in our primary response. So what interferon do, as it says here, it will activate an antiviral state in the following cells. So when this virus goes to the next cell, this cell is essentially ready because it's been stimulated by interferon gamma. So this is where it enacts its functions of here, as you can see here. So it essentially communicates with other cells to be prepared and recruits other cells, as we'll get into later. So we also have molecular defenses through the interferons. So our type 1 interferons, which are alpha and beta, are produced by cells in response to viral infection. The type 1 interferons act to protect neighboring cells, as we discussed before. So interferon production is triggered by viral replication in the initial cell, and these interferons are released by this infected cell. The interferon binding to the interferon alpha receptors on neighboring cells induces antiviral protein production in those cells. The original infected cell will then die, as we can see here. We can see up here, the cell now dies. You can see it leaking out. Then the virus-infected cells will activated by interferon to produce AVPs. These AVPs are active in the other cell, and the AVPs degrade the viral mRNA, bind to ribosomes, and block viral transcription and translation. So, our action of type 1 interferons. So again, these type 1 interferons bind to a shared interferon receptor, the interferon alpha receptor. This binding induces activation of transcription factors, including STAT1, STAT2, and IRF9. This results in assembly to form the complex ISGF3, which is also, as we said earlier, is interferon-stimulated gene factor 3. ISGF3 translocates to the nucleus, and this activates transcription of interferon-stimulated gene, or ISG. So here is a good summary of the terms, if you want to just catch up on some of the acronyms as I'm going through. So again, there's 3 key roles for the type 1 interferons. They induce resistance to viral replication in unaffected cells by inducing expression of interferon-stimulated genes. They encode antiviral proteins, or AVPs, leading to the destruction of mRNA and inhibition of protein translation. They can also activate NK cells, and these kill virus-infected cells, and they can also induce MHC, or class 1 MHC expression, which we'll discuss next. So we can see here, it can induce class 1 MHC expression and increases susceptibility to cytotoxic T lymphocytes. So as we discussed in the first lectures, if we have increased class 1 MHC that is presenting virus, CDLs or CD8 T cells bind to MHC1. So it kind of works together. So by inducing this, the interferons can increase this and then help the adaptive immune system identify these targets. So here we can say that the interferons also induce resistance to viral replication in unaffected cells. So this is because they induce expression of interferon-stimulated genes, which encode these antiviral proteins, leading to the destruction of mRNA and inhibition of protein translation. So an example of these antiviral proteins include oligodenylate synthase, PKR, or protein kinase that inhibits the translation by binding to EIF2, and IFIT proteins which suppress viral RNA translation. The net effect of antiviral proteins is to inhibit viral synthesis and limit the spread of the virus between cells. So here's pretty much the summary figure, which we haven't covered all of these yet, but it's just a good way to see. This initial cell that's infected releases alpha and beta interferon, and because of activation of our transcription factors, increased viral resistance to viral replication, increased expression of IFIT, which we'll talk about later, increased MHC, as we just discussed, increase in our antigen-presenting cells, increase in NK cells, and an increase in chemokines to recruit lymphocytes. So now we're going to talk about each of the antiviral proteins in a little more detail. So first we have oligodenylate synthase, or synthetase. This is activated by double-stranded RNA during viral infection. So active adenylate synthase polymerizes into a 2' 5' linked oligomers. So as you know, you may have learned our DNA is 3' to 5'. So this is an atypical linkage, and this activates an endoribonuclease called RNase L, and this degrades viral RNA. So these interferon-induced AVPs sense the difference from our normal 3' 5' nucleotide linkage in nucleic acids, which results in viral DNA degradation. So now we're going to take a look at PKR. So protein kinase R, or PKR. This also has a quite complicated name, also known as eukaryotic translation initiation factor 2 alpha kinase 2. PKR is a double-stranded RNA-dependent protein kinase able to inhibit translation. So type 1 interferons can induce PKR synthesis. Viral double-stranded RNA binding to PKR results in the active PKR through autophosphorylation. So active PKR then phosphorylates and inactivates EIF2. Then EIF2 is needed for translation for the formation of the pre-initiating complex. So viral proteins production is stopped, and the normal host cell protein synthesis is also inhibited. So, once the action of interferons, one action of interferons is to induce the PKR synthesis, as we said. PKR is activated when it binds to double-stranded RNA produced during viral replication. So, importantly, inactive PKR then inactivates EIF2, shutting down protein synthesis for both the virus and the cell. So now we're going to take a look at the IFIT proteins. So the IFIT proteins, also known, that stands for interferon-induced protein with tetratrichoid repeats, are antiviral proteins that inhibit early steps in RNA translation, and they block viral protein production. So in a virus-infected cell that has been activated by type 1 interferons, the IFITs can attach to eukaryotic initiating factor 3, or EIF3. So EIF cannot interact with the other components of the 43S complex. So this results in translation being inhibited in the ribosome, so no new viral proteins are synthesized. So again, here's some additional, so these we already covered. So just kind of a quick review. And again, take a look at this figure if you want a little more detail and follow the sequence. But there's also some additional roles for type 1 interferons, and this includes the activation of dendritic cells and macrophages, and chemokine induction for lymphocyte recruitment to attract cells to the area of infection. So now we're going to look at natural killer cells in a bit more detail. So NK cells help limit virus infections while adaptive immune responses prepares. The type 1 interferons are produced early in virus infections, and these type 1 interferons increase NK cell activity through interferon gamma. Alpha and beta, IL-12 also increases NK activity and induces NK cells to make interferon gamma. Now, interferon gamma released from NK cells results in macrophage activation. The adaptive immune defenses come in later, and this includes cytotoxic T lymphocytes, which kill virus-affected cells, and virus-specific antibody, which can cause neutralization, opsonization, and promotes ADCC. Essentially, interferon gamma or interferon-induced NK cell proliferation and killing activity as an innate response buys time for the adaptive immune response to act. So here you can see here our initial response, production of interferon alpha, beta, TNF alpha, and IL-12. Then we have our NK cells recruited. This essentially buys us time until the adaptive immune response can come in and get rid of the virus. So again, interferon alpha and beta induce the production of antiviral proteins that inhibit further virus production. They induce increased expression of MHC class 1 molecules on host cells, which allows CD8 or cytotoxic T cells to more effectively see their targets. This also results in an increase in natural killer cell activity, and these NK cell activity increases type 1 interferons, which are present. The result is that the virus that induces interferon induces NK cell activity, and so induces killing of the virus-affected cells. So here's a really good figure that kind of sums up the whole process. So we see here, interferon, NK cells, enhanced killing, enhanced MHC expression, enhanced activity of our CDLs. We also have direct degradation of viral DNA, as we talked about earlier, blocking replication. We also induce our antiviral proteins, like we said, oligodenylate synthetase, PKR, and those. And we also, which results in inhibition of viral protein synthesis. So again, just a quick summary. We got our interferon alpha and beta here, and our three roles we discussed: resistance to replication, NK cells, and class 1 MHC. So now we're going to take a more detailed look at NK cells in this section. So again, quickly, NK cells are activated by interferons and macrophage-derived cytokines, which we'll talk about later. As you can see here, one hint there, to serve as an early defense against certain intracellular infections. So, NK cells are a type of lymphocyte and are closely related to T cells. NK cells are now grouped in the group 1 ILCs, which we discussed in an earlier lecture. NK cells are key players in innate immunity, especially against viruses, and they play a major role in cytotoxic activity. But they're not as specific as are CD8 T cells, and they do not require prior exposure to the target, and they have no memory. They're activated by the type 1 interferons, alpha and beta, as well as IL-12. These cytokines can result in a 100-fold increase in activity of NK cells. So NK cells kill virus-infected cells, and some tumor cells are targets as well. Some can say it's more cells that are experiencing stress or damage, which can kind of happens in both these scenarios. NK cells and both CDLs use similar methods of killing target cells, and they both destroy their cells using granzymes and perforin. So again, we've discussed this before, so I won't go over it quickly, but just remember, these are now in group 1, and the innate cells are also not specific. So NK cells recognize their targets through different kind of mechanisms, but it's also important why they don't kill our own cells, as you can see here. So NK cells contain inhibitory receptors, which is sometimes seen in textbooks as KIR, as killer inhibitory receptors. So these inhibitory receptors recognize class 1 MHC. So as we discussed, class 1 MHC is pretty much on all cells of our body, and it's almost kind of like a self molecule. So it basically sees class 1 MHC and knows that we shouldn't be killing that cell. So as you can see here, if NK cell sees class 1 MHC on the cell surface, NK cell killing activity is inhibited. Class 1 MHC proteins are constitutively expressed on normal host cells, so they are not killed by NK cells. So here we have our inhibitory receptor on our NK cell, comes into contact with a cell expressing the normal amount of MHC, this is kind of little drawings. It will receive the stop signal and no killing will occur. On the opposite side, we also need to wait for them to actually know when to kill cells. So we have our activating receptors. So these activating receptors are recognized stress-associated molecules. These have also been seen in the literature as KAR, killer activating receptors, but they also have other names, which we'll get to later. So these stress-associated molecules are expressed on virus-affected cells and some tumor cells or damaged cells. Examples of these stress molecules include MIC-A and MIC-B, and we can see that MIC-A stands for MHC class 1 polypeptide related sequence A or B. So if an activating receptors on the NK cell sees the ligands on the host cell, this signals the NK cell to kill the target. There's an important need for balance between inhibitory and activating signals. So we can see here an NK cell interacting with an abnormal host cell. The interaction of the activating receptor with stress proteins on the host cell sends an activating signal to the NK cell. So this cell is abnormal. Maybe it's a tumor cell undergoing rapid division. Our body expresses MIC-1 or MIC-A, or MIC-B. Our activating receptor recognizes, initiates the kill signal. Interestingly, there's also another way this works, is through the fact that some viruses decrease the class 1 MHC expression on affected cells. So as we talked about earlier, we have the KIRs, which looks at MHC1. So all of a sudden, your MHC goes down. There's no inhibition of that signal to kill. So activating receptors recognize these stress molecules, like we said. So if there's no MHC1 to stop this, to initiate the inhibition, it will proceed with killing. So again, NK cells have inhibitory receptors for class 1 MHC, KIR, killer cell, also known for killer cell immunoglobulin-like receptors. If the IRC class 1 MHC on the cell surface, the NK killing activity is inhibited. And again, normal host cells express class 1 MHC and are therefore not killed. Killing by NK cells depends on the balance. Again, in immunology, balance is always important between activating and inhibitory signals. The interactions of inhibitory receptors on NK cells with MHC class 1 proteins on target cells sends an inhibitory signal to the NK cell and inhibits killing. However, the inhibitory signal is lost when target cells decrease or lose MHC class 1 proteins, which occurs, as we said, when some viruses infect our cells. So basically, it's a way that gives away the virus's location. So NK cells can kill more host cells with reduced class 1 MHC expression, or have expressed the MIC-1, MIC-A, and MIC-B receptors. So again, if class 1 MHC expression is altered or decreased, NK cells will still kill the cell. So if you have a loss of signal transmitted through the inhibitory receptor, this will kill the cell. NK cells can kill host cells with abnormal expression of class 1 MHC. And this strategy is also used by some viruses to prevent NK cell killing of infected cells. So some cells can actually preserve the MHC class 1 repertoire, preventing being killed. But in a lot of cases, some viruses will block class 1 MHC protein transport to the cell surface from inside the cell. And this means infected cells have decreased class 1 MHC expression, again being targeted for killing. And some viruses also block whole cell protein synthesis and cause decreased class 1 MHC expression. So again, we'll take a closer look at these inhibitory receptors. So how do they work? So these inhibitory receptors have long cytoplasmic tails with ITIMs. This stands for immunoreceptor tyrosine base inhibition motifs. So there's ITIMs and also ITAMs, as we'll see later. The I is for inhibition, A is for activating. So this is just the easiest way to basically the easiest way to memorize what each one does. ITIM, you look for the I, inhibition. So the ligand binding to this inhibitor receptor results in the phosphorylation of the tyrosine in the ITIM. This binds to the intracellular phosphatases, removes the phosphates from the tyrosine residues of other signaling molecules, and inhibits cell signaling, and no killing occurs. So as we see here, certain KR are inhibitory receptors, while others are activating receptors. Their actions reflect the presence of ITIMs or ITAMs. So again, inhibition, activating. So now we're looking at target recognition. So NK cells also sense ligands on virus-infected cells and tumor cells. They look for those stress-associated molecules like we talked about. These are sensed by NK cells through activating NK cell receptors or NCRs. These activating receptors transduce an NK activating signal. So activating receptors have short cytoplasmic regions and interact with transmembrane signaling proteins including DAP12, zeta, and zeta, which have ITAMs. As you can see here, ITAMs depicted as yellow rectangle, which you can see is on these different cells, both on zeta and DAP10 and DAP12. And these motifs participate in signal transduction. So we can see here NK cells can kill virus-affected cells and tumor cells due to the expression of ligands induced by cell stress. So here are a bunch of different stress-associated markers. So these receptors are shown here. And again, ITAM stands for immunoreceptor tyrosine base activation motive. We can see here that DAP12 has ITAMs as well, and DAP10 activates the kinase that increases NK cytotoxic activity. So NK cells can sense this danger to the host by recognizing these ligands induced by the cell stress. This can be caused by viral infection, intracellular bacterial infection, and malignant or transformed tumor cells. These ligands include our class 1-like molecules, which were MIC-A and MIC-B, like we talked about earlier, and also RAYIT1, which has similar portions to the class 1 MHC. So NK cells can kill virus-infected cells and tumors due to expression of ligands induced by cell stress. Examples of these ligands are shown here in MIC-A, MIC-B, and RAYIT1, as we said. And the RAYIT family of ligands are for the activation of receptor NKG2D. So how does this activating receptors work? So these activating receptors have short cytoplasmic tails with ITAMs. Again, activation motifs. These ligands binding to the activating receptors causes phosphorylation of tyrosine in the ITAM, causing intracellular signaling pathway activation. The NK cells release their cytotoxic granules and the target cell killing occurs. Again, we got a different set of molecules here. And now we have our ITAM instead of ITIM. So another way of NK cell function is through the trail-mediated actions. So the trail-mediated interactions are another strategy for target cell killing by NK cells. So trail is from the tumor necrosis factor receptor family. So trail stands for a tumor necrosis factor related apoptosis inducing ligand. So it kind of gives its function in its name here, inducing ligand, apoptosis inducing. So trail, which is on NK cells, interacts with a cool receptor, at least name-wise, called death receptor on target cells. The binding of trail to death receptors induces signaling pathways on the target cell, causing apoptosis. So again, death receptor kind of makes sense for its name. If that gets stimulated, still the cell will die. So we'll go into this detail here. NK cells can also kill their target cells through interactions of trail, again on the NK cells, seen here, with members of the TNFR or tumor necrosis factor receptor family, including death receptor 4 and 5. You can see here on target cells. So when trail binds to these death receptors, a signal transduction pathway is induced in the target cell that leads to the caspase activation. So we can go from pro-caspase to active caspase. This causes the induction of apoptosis in the target cell. We can also have the activation of FAD, or FADD, known as fast-associated death domain. You may see in a lot of other literature, so FAST is pretty much involved in apoptotic pathways. So many cells can also induce this path, the FAST ligand. Even some viruses can cause death of our cells through FAST. And this is an adapter molecule in this. So again, FAD in this pathway is an adapter molecule, and it activates procaspase 8 and also causes the protease cascade and apoptosis as well. So again, what does NK cells do to their target? They kill these target cells through apoptosis using their perforins and granzymes. So perforins create pores, kind of makes sense in their name, create pores in the target cell membrane, while granzymes can enter the target cell and induce apoptosis. And we also have caspases, which are a family of cytoplasmic proteases that cleave key cellular components. All of these result in damage to the cytoskeleton of the cell, its DNA, its chromatin, and causes cell death. So again, target cell dies by apoptosis. So it's important to remember that apoptosis is an organized cell death compared to necrosis or pyroptosis that is more harmful to the host. This is targeted and very strategic. And then you can see here our macrophages will come up and clean the debris after apoptosis occurs, eating the apoptotic bodies. So during apoptosis, we have chromatin condensation, the cell compacts, and the membrane blebs or apoptotic bodies form, which are then phagocytosed by macrophages, as seen in the figure. So again, the roles of NK cells are to kill infected target cells, produce cytokines which affect other cell types. So again, NK cells make interferon gamma, which activates macrophages, which cause increased killing of pathogens, as we can see here. Then we can also have macrophages and NK cells, which work in synergy. These macrophages make IL-12, which activates NK cells, and these NK cells make interferon gamma, which activates macrophages. So they kind of work together. That's why you can see this loop as a circle, essentially. Essentially, it's a positive feedback loop, but it's not, and it's still under control. So there's also more synergy. NK cells can use our antibodies to kill target cells as well. So this process is antibody-dependent cellular cytotoxicity, or ADCC. So this works by this is effective after antibody is produced. So antibody can attach to the surface of infected cells, and the Fc portion of the antibody, as you can see here, the stalk of the Y, is then seen by our NK cells, which have Fc receptors which bind to these antibodies with higher affinity, and it's more easy for them to target, and the NK cell kills the target cell. So here's just, we'll go step-by-step process. So first, the Fc receptors recognize the conserved portion of the antibody molecules. So Fc receptors on the NK cell bind. You can see that here. The Fc receptors expressed on the surface of the NK cells allows the NK cell to attach to the antibody-coated target cells. The infected target cells recognized in this way will be killed by the NK cell using ADCC. So again, if you want to take another look at this figure, this is a good figure to take a closer look at. So again, ADCC is antibody-dependent cell-mediated cytotoxicity. So here we have a little animation we can follow here. Antibodies binding to the cell. Here comes our NK cell, as we saw the MC portion, attaching to the Fc receptor. Now the NK cell releases perforin and granzymes, and then these will degrade the viral protein, causing apoptosis. So again, we have our Fab portion of the antibody, which is these will bind to the epitope on the target. The Fc portion of the antibody binds to the Fc receptor on the NK cell, and the NK cell gets close to the target and releases perforin and granzymes, causing apoptosis. So again, we have a nice summary figure here, like we did with our interferons. So we have our NK cells, which kill abnormal host cells that have decreased class 1 MHC expression. These NK cells also kill infected host cells by using ADCC, as you can see here. They also have synergy with the interferons, because NK cells produce interferon gamma, which activates macrophages, and macrophages in turn activate NK cells through IL-12. Many viruses induce type 1 interferon production by infected cells by the release of interferon alpha and beta. These NK cell activity increases type 1 interferons are present, and the result is viruses that induce the interferon induce NK cell activity and so induce their own death. So yeah, and again, something that's no show's finger is the newer pathway discovered. It was the trail-mediated interactions with death receptor on the target cells, which also induce apoptosis through Cas-based pathways. So, also important to keep in mind is how do we see the virus. So we also have PRRs. So PRRs are key innate defenses against viruses, which include NK cells and interferons. We also use PRR-mediated recognition of virus components, which causes the activation of macrophages and dendritic cells. So how are PRRs involved in sensing viruses? So TLR-mediated recognition of extracellular viral RNA and DNA is taken up by endocytosis. And these are sensed by these ones, as you can see here, TLR3, 7, 8, and 9. These recognize the viral DNA components and RNA components, compared to these ones that tend to detect bacterial components in extracellular spaces. We also have RIG1-like receptors, or RLRs, that sense intracellular viral RNA. And this is RNA synthesized in a virus-infected cell. And we also have STING, which is stimulator of interferon genes, which works with C-gas to sense cytosolic DNA, including double-stranded DNA from double-stranded DNA viruses and bacterial DNA. So we can see here that the TLRs recognizing viral nucleic acids are located in the endosomal membrane, as we can see here, endosome. And these detect viruses entering by endocytosis. So we've kind of go over some of this stuff with signal transduction in our other lecture, but we'll just quickly go over it with the fact that we have the common endpoint of the activation of NF-kappa B, causing transcription and the release of these cytokines, including IL-1, IL-8, and IL-6. And again, we have our TLRs, NOD1, NOD2. These all work to activate NF-kappa B. So again, here's a figure from last week, just a summary of what each TLR does and which pathway it goes. We can kind of see here, so as we talked about interferon, these are also involved in interferon production. So even though these are different pathways, they still result in these pathways that end up in the release of our NK cells and other antiviral components. So as we can see here, signaling induced by TLRs involves sequential activation of these protein kinases and adapter proteins and scaffold formation, ultimately leading to the activation of the transcription factor NF-kappa B, as we've discussed. But some TLRs use the IRFs, like TLR3 and 7. Working together, the TLRs can activate the transcription factor NF-kappa B, AP1, also known as activator protein 1, and IRF, interferon regulatory factor, to induce the expression of inflammatory cytokines and type 1 interferons. So another example of this is our LPS and TLR4 signaling. TLR4 uses help from accessory proteins, including MD2, CD14, to bind to LPS. This signal is induced by TLRs, involves sequential activation of protein kinases and activator proteins and scaffold formation, ultimately leading to activation of transcription factors NF-kappa B. Oops. So again, common steps in TLR signaling. Our TLR ligands bind to our TLR. TLR dimerizes. It recruits adapter molecules, including MyD88, to the TLR complex via TIR domains. And the MyD88, again, is an adapter protein. So MyD88 recruits protein kinases, including the IRACs, which recruit and activate ubiquitin ligases, which includes TRAF6 and others, resulting in polyubiquitin chains. So the next steps in TLR signaling, this results in the production of a scaffold for the activation of the kinase cascade. This recruits adapters and kinases. These adapter proteins, or tabs, bind to the scaffold in TAC1. This results in TAC close to the This brings TAC close to the IRACs. The result is that the IRACs phosphorylate TAC1 and activate the kinase. So we can see that here. IRAC becomes close to TAC1, activates the kinase. Then this active TAC1, as we can see here, phosphorylates IKK, which is the I kappa B kinase complex. IKK is a complex of 3 proteins, IKK alpha, beta, and gamma. And IKK gamma is also known as NEMO. And NEMO binds to this polyubiquitin chain. So the last steps, we have the activation of IKK by TAC1. So NEMO and IKK gamma binds to polyubiquitin chains. The scaffold brings IKK complex close to TAC1. TAC1 phosphorylates IKK beta. IKK beta phosphorylates I kappa B, which is the inhibitor of NF kappa B. As we can see here, with this current situation, NF kappa B is inhibited and won't move to the nucleus. Once I kappa B is degraded and removed by IKK beta, it allows NF kappa B to be free and move to this nucleus to transcribe cytokines. So again, not all TLRs use the same adapter proteins. TLR3 uses IRF3. TLR7, which senses single-stranded RNA, looks at IRF7. Uses the transcription factor IRF7, sorry. So you can see the pathways are slightly different. You can see our 2 different unique pathways. So again, a good way to sum this up is our TLR interactive pathway on this website here. Again, check it out if you want to follow in detail. So now for some more detailed information. So we just needed a little bit of background on those TLRs to get to this. So now we can look at STING and cGAS, which sense cytosolic DNA and cytosolic dinucleotides. So STING, known as stimulator of interferon genes, is a cytosolic sensor of DNA, of double-stranded DNA, looks at bacterial DNA, double-stranded DNA from double-stranded viruses. These sense cytosolic dinucleotides. These are made by bacteria or induced by viruses via cGAS. So if STING sees cytosolic dinucleotides, it will cause a conformational changes which recruits kinases such as TPK, which activate transcription factor interferon regulatory factor, which again results in our interferon 1 expression in all the pathways we discussed earlier. So essentially, this activated IRF3 will go into the nucleus, act as a transcription factor, release type 1 interferon genes. So viral DNA bound to cyclic GMP synthase, or cGAS, results in the production of cyclic GAMP. So we go from cyclic GMP, AMP, from ATP to GTP, and cyclic GAMP activates STING. So as we discussed a little bit earlier was the RIG-like RIG-1-like receptors. So just to overview here, the RIG-like receptors sense viral RNA in the cytosol. The RLRs can interact with adapter proteins MAVs with help from the ubiquitin ligase TRIM. Ubiquitin scaffold formation results in the kinase recruitment, and then again activation of NF kappa B and pro-inflammatory cytokine expression. We also have activation of the interferon regulatory factor, IF3, resulting in more type 1 interferon expression. And again, all of these are focused on antiviral actions. So again, senses that viral RNA. Then we have our RIG1 pathways, MAVs, which is our adapter protein. You can see the mitochondria. Then again, we have these formation of these viruses bound to the receptors. Then we have the formation of our transcription pathway. And then again, final steps. I kappa B removed from NF kappa B for cytokines. And IRF3 to help release interferon. So just a quick way to bring it back to COVID. So again, we saw in here, we can see MDA5 stuff like that. So a lot of those are involved in these transcription factors. So again, MDA5 governs the innate immune response to SARS-CoV-2 in lung epithelial cells. So these pathways, although may seem a little boring and a little complex right now, are relevant to current situations. So here we can see MDA5 in the same pathway here. So we have our IRS and our NF-kappa B. Again, here's another paper, or here's the highlights of the paper. So SARS-CoV-2 replication induces delayed interferon response in lung epithelial cells. MDA5 and LGP2 are the major sensors of recognizing SARS-CoV-2 infection. Viral intermediates activate interferon response through MDA5 mediated sensing, and IRF3, 5, and NF-kappa B are required for interferon response induced by SARS-CoV-2. So in the end, you can see that basically all this is to help us fight viruses. And this is technically innate. So we're not even talking about what T cells are doing after this is happening, but these are the processes that are essentially keeping the viruses at bay while their adaptive immune response develops. That's why interferons and NK cells are so important. And again, we can see here, bacterial coronaviruses have varied strategies to evade innate host defenses. So we can see here, severe SARS-CoV-2 the M protein inhibits type 1 and type 3 interferon production by targeting ring 1 MDA5 signaling. So essentially, the virus is able to disable our innate responses, because essentially, if these innate responses could work at 100% efficiency, we might not even get sick. So clearly, the SARS-CoV-2 is disabling our system to survive. So we can see here that ring 1 and MDA5 are being targeted by the virus, affecting our interferon production, allowing the cell to replicate with much more ease. Essentially, it's not able to keep it at bay as well as it should, but we can still have our adaptive response come in. But sometimes it can be too late. So here we can see the negative outcome of suppressed or delayed interferon production on the responsive virus infection. That's the end of the lecture, guys. Hope you enjoyed it. I know it can be a little dry at some points, but I hope you learned some stuff. Thanks. Take care.